1 in 5 women will not be able to conceive a child naturally. One can be young, fit, and generally healthy, yet your eggs may still not be of a sufficient quality to support a healthy pregnancy.
We aim to deliver the biggest improvement in fertility since the introduction of IVF. If successful, EmbryoProtect could become the lead in fertility therapy of the next decade, with revenue potential comparable to the top 30 best-selling drugs globally.
Whether a pregnancy succeeds or fails often comes down to one thing: whether a single cell gets its chromosomes right. At the Max Planck Institute for Multidisciplinary Sciences in Göttingen, we spent nearly a decade trying to understand one thing: why so many eggs fail. The egg is the most important cell in the human body, and it carries everything needed to start a new life. For a long time, the field focused on hormones or lab techniques because the egg itself was simply too difficult to study. Only recently we've been able to look inside and understand what actually goes wrong. What we discovered in my lab is that as a woman ages, the chromosomes in her eggs fall apart. And we were really surprised by how many eggs were affected. The reason is a molecular glue that holds the chromosomes together but gets weaker over time. As a result, chromosomes can fall apart or separate too early. And we identified a second problem. The spindle, the machinery that pulls chromosomes apart, is not perfectly reliable, even in young eggs. These two mechanisms together are the reason why so many eggs end up with the wrong number of chromosomes. For a long time, it was simply not possible to observe living human eggs in detail. We developed methods to safely image them and watch chromosome behavior in real time. This work began while I was a group leader at the LMB in Cambridge. The Max Planck Institute then provided the environment, expertise, and long-term support to scale up this work, uncover the mechanisms that lead to errors, and ultimately explore ways to reduce them.
I still very much remember my first meeting with Melina in 2013. What immediately captivated me to join her lab was the opportunity to study real human eggs donated by patients and not animal models.
There was a moment when we realized that the mechanisms we discovered were not just descriptive, but actionable. If chromosome instability is the core problem, then stabilizing chromosomes could directly improve egg quality. That was the turning point.
What makes our approach unique is that for us, the egg itself is the patient. The treatment is applied directly to the egg in the lab after it has been retrieved as part of a standard IVF cycle. And because everything happens outside the body, it adds no additional burden or side effects to the patient. In simple terms, the magic happens inside the dish. Our EmbryoProtect platform targets two key aspects. First, chromosome integrity. Over time, the glue that holds chromosomes together weakens. We address this with EmbryoProtect 1, which stabilizes the natural glue, and EmbryoProtect 2, an engineered version for more advanced cases. The second mechanism is the spindle. With EmbryoProtect 3, we can make chromosome movement more efficient and reduce error rates. The one constant at Ovo Labs is that no 2 days are the same. There is always a moment of anticipation when we open the incubator and see how many research eggs there are and their age. And then the real work begins. One of the first things that might surprise you is that we give every human egg that passes through our hands its unique identifier. If you say to me human egg 183-1 from Munich, I can see in front of my eyes what has happened to it and how good or bad its chromosomes looked. We use complex instruments to deliver minute volumes of the drug in the picoliter range with unprecedented precision, and then closely monitor each egg as it segregates its DNA using advanced microscopy techniques. And at the end of the day, the moment of truth, scoring how healthy the egg is and to what extent we are able to positively impact its trajectory.
The idea originally came from Melina, who wanted to take her research beyond academia and translate it into real clinical impact. She partnered with Agata, who was a natural choice. Together they discovered the mechanism behind poor quality some 10 years earlier that Ovo Labs is now focusing on. And as with many good things in life, Ovo Labs came into my world quite unexpectedly. I was introduced through a mutual connection, and it clicked very fast. From a purely rational perspective, leaving my previous career did not make much sense. But I wanted to build something meaningful that has a positive impact on life of others. Through the spin-off process, Max Planck benefits financially from the success of the company, while we receive exclusive global and perpetual access to the patents that define our EmbryoProtect pipeline. We received strong support from Max Planck Innovation, who work closely with us as true partners throughout the process.
Ovo Labs is a great example of how long-term basic research can translate into an application. The EmbryoProtect pipeline was built on many years of fundamental research in my lab, and through this work we could now develop a completely new therapeutic approach.
If we are successful, long-term we could enable birth of up to 1 million babies each year that otherwise wouldn't have been born. And that, of course, has a massive societal and economic impact, especially in today's world where birth rates are falling off the cliff. What makes this scalable is that IVF is a highly standardized procedure. Clinics around the world follow similar processes, and we have already established partnerships with more than 10 IVF clinics around the world, including one of the largest clinic networks.
If we succeed, it could give women real reproductive freedom, especially in a world where family planning often happens later and at the time where fertility is already suboptimal. The scale of this societal impact is exactly why I chose Ovo Labs and our mission beyond anything else.
What really fascinates me are the many moments when you see something that no one has seen before. That feeling of discovery is incredibly powerful. And now there's an additional motivation, knowing that this work could actually help patients, and that combination really keeps me going. At the same time, the research at the Max Planck Institute will continue to evolve and feed into the next generation of therapies.
I am a father, and parenthood has been by far the most meaningful experience of my life. Before Ovo Labs, I had not thought much about fertility, but when I first heard about the idea, it really captivated me, and it made me realize how many people struggle to experience something that I'm very fortunate to have. In 5 years, I hope we will see the first babies born following our treatment. The Gründer's Prize is the beginning of the next phase for Ovo Labs.